The main aim of this procedure is to provide a cost-effective and rapid molecular genotyping protocol that can differentiate between varieties of Imperata cylindrica, typically called cogongrass, that cannot be distinguished by morphology alone. This is accomplished by first identifying and isolating tissue from a cogongrass variety of interest. The next step is to extract nuclear and plastid DNA from the collected tissue. Next, polymerase chain reaction is performed on the extracted DNA using variety-specific primers that target sequence differences in the plastid trnL-F region, as well as appropriate positive and negative controls. The final step is to perform gel electrophoresis on each of the 4 PCR reactions to visualize the amplified PCR products. Ultimately, the variety of cogongrass collected is determined through analysis of the resulting variety-specific PCR product bands on the gel.
Invasive plants cost the United States about $3.4 billion annually, causing substantial harm to both agricultural and natural resources. Wild-type Imperata cylindrica, also known as cogongrass, is one of the top 10 worst invasive weeds in the world. Cogongrass forms rapid-spreading monodominant stands that displace native vegetation and in turn the animals that depend on that native vegetation. Cogongrass currently infests 490 million hectares worldwide and about 500,000 hectares in the United States. An ornamental variety of Imperata cylindrica is widely marketed under the names of Imperata cylindrica rubra, red berry.